All right, hello, Advanced Lab. So what this video is I'm talking about the lab report assignment that you have to do. So part of this class, and if you watch the syllabus video, part of this class serves as your Tier 2 writing credit for Tulane. And so because of that, there's a large component of writing. So if you go to the lab reports down here, there's a Lab 1 paper. It's not visible yet, but it will be for you guys. And so we'll click on it, and this is just briefly the assignment. And this is where you're going to upload it. You're going to upload a PDF. It should be about 5 pages long. Could be a little bit more. That's okay. That's not like a strict thing. And what I'm going to walk you through is how to actually, what I'm looking for in terms of the paper and the structure and all that. You've already gotten familiar with using LaTeX, so you'll use that to write the paper. That already had some sections in it that you can use and some examples of equations. So let's look at the PDF here that I've created for the instructions. So this is the written lab instruction. I'm not going to read this verbatim because you can read it yourself, but I'll go over it in general. So this just talks about creating the paper. It's going to have to be in LaTeX or LaTeX. And the figures will also be from Python and MATLAB. You can't use Excel figures. This is kind of the standard. If you're in a research lab and you were doing a group meeting presentation, you wouldn't show people Excel, or maybe you wouldn't have group meeting, but you wouldn't do it in a like a presentation at a conference or anything like that. So I'm getting you guys used to kind of being professional about it. All right, so this is the overall structure of the paper. Again, this is mirrored in the outline I gave you before. So the first is the title. I say give your paper a good title here. Superconductor lab is not a good title. You want to describe actually what it is. So we don't have it here, but there can be an X-ray diffraction lab where you use X-ray diffraction to analyze structures of materials. So it might be analyzing structures of various materials via X-ray diffraction. Or if it's something more specific, analyzing the structure of copper powder, or something like that. That would be too. But titles like one short phrase or sentence. Then you're gonna have the abstract underneath that. The abstract is a concise summary of your report. It's what you did, how you did it, and the results. So it says 200 words or less. That's kind of a guideline. Again, I'm not like the word police. If you have 220, I'm gonna come get you. But that's just a guide. And I have here, it's actually the most important part of a scientific paper. Paradoxically, it's actually often written last. You would do the abstract after you've completed all the rest of the analysis, and sometimes it takes writing the whole paper to kind of figure out how you're going to frame the research you're doing. And so often abstracts are written, submitted alone. So if people are looking for your paper on websites like Google Scholar or anything else, often they'll be able to just see the abstract. So the abstract has to kind of stand alone. So you can't reference figures or equations or citations or use acronyms. It just has to kind of stand alone. So the abstract is kind of like a summary of what the paper is. When I've written papers in the past, you spend untold days just working on the abstract. So it's super important. So I'm just telling you this, like I will be looking for a decent amount of effort being put into the abstract. The introduction is the beginning of the paper. This is usually about 2 paragraphs, and this just motivates history. In some ways, the introduction is really important in terms of getting the reader familiar with what you're talking about, and in some ways, it's the least important part of the paper because it's kind of a standard thing for different topics. You're kind of motivating what you're doing, what's the history of it, and that kind of thing. You're not going to put really any equations or plots. You might put a figure in there if it's like useful. I'll show you an example in a minute where you do that. After this comes the theoretical discussion. This might be blended with the introduction. You can think about like the theory and the introduction of your thing. Like say you're doing the photoelectric effect, you would talk about Einstein's ideas. It's actually an opportunity you can actually reference Einstein if you're doing that presentation or that topic. And this is where you would give the theoretical baseline of what you're working on. Because the experiments here, we're not developing new theories, the theory is kind of like background as well. So you can have figures here as well describing, say, the photoelectric effect again, where the light comes in, it hits a metal and releases electrons. You might put in a picture of the circuit or something like that. But you'll put this here. This is not always how papers are written. I'll show you an example. I'm going to show you one of my papers from grad school, and I didn't have a theory section. That's not always shows up. All right, then you have the experimental method section, and I actually think this is a really important thing because we're doing experimental work. You put a diagram in here of the stuff you're doing. Basically, I think this is the most important line here. It contains enough detail so that one of your peers could basically set up and do the experiment using the paper. So you should be concise enough that you describe exactly how to do it, but don't go on for too long and make it really long. You're going to be tempted for the section to put in like a bullet point list. You don't want to do a bullet point list. The entire paper is written like a narrative, so it needs to be a narrative. Then you have the data analysis and results. Just like your lab notebook, the data analysis and results is the most important part. So this is what you spend the most amount of time on. I even bolded it here. This is where you put your data and your analysis. Is where all the plots are going to go with the fits and the information extracted from the fits. If there might be equations here where you relate the pure theory to the plots you have and how you combine this to get the information you want, you put that here as well. I would like it if everybody put in a chi-squared analysis so you can tell me if your fit's good or not. And this is a good opportunity to discuss that. And again, this is written as a narrative, not a list of results, but like describing basically, again, what did you do, what did you get, and what were the results, and what does it say? And at the very end, I actually think this wasn't in the template I gave you, but at the very end, the conclusion is just a one paragraph that summarizes the big picture of the results of the experiment. Often, conclusions can be a place where you put like next steps. What would you do if you had to redo this experiment or you had different equipment? Like what would be kind of your next steps? After that come the citations. The citations are referenced. If you look at the example I gave you, the template, the citations, you list them earlier. The citations will mostly show up, honestly, in the introduction and the theory because you're referencing previous work, but then it'll show up at the end of the paper. I'm saying here you need at least 2 citations. Here are some other tips and tricks, things I think are important. Basically, figures and writing. Figures are the second most important part of the paper behind the abstract. Often when people are reading scientific papers, what they do is they read the abstract. If that looks interesting enough, then they'll go through and look at the figures and read all the figure captions. And that might be the only thing they read about the paper, even if they think it's kind of interesting and it's kind of good work. If they really think it's relevant for what they're doing, then they'll read the rest of it. But the rest of it is kind of last. So people do not read scientific papers beginning to end. They kind of pop around. So the figures are the second most important thing. So that way they should look good. You need to have correct legends and units on everything. It needs to be good size. It can't be overflowing the 2-column format. You should have, I say here, structures of plots vary based on experiment. But in general, data is represented by points and fits are lines. And don't have, if it's a plot where it's just like a bunch of data in one line, don't put a legend in there that says data fit because it's obvious what's the data and what's the fit. The legends are necessary if you have like multiple lines on this. Oh, this is the green curve and this is the yellow wavelength curve and this is the red one. Then you maybe need a legend. Well, you do, but not if it's just one data, one plot. And then I say here, the figure caption contains the relevant information. You can put the fitting parameters in here. You can say where the fit comes from again, because people are going to read the figures outside the context of the rest of it. So that kind of helps you, the figure caption kind of helps you hold the figures kind of standalone. They can be one sentence and they can be a couple of sentences. I'll show you an example where I have really long ones. Writing tips, these are really important. I'll be a little nice on this in the first time, but this is how you write scientific papers, at least currently. So don't use the passive voice. What does passive voice mean? The experiment was performed. It acts like it's passively done. You want to use the active voice. We performed the experiment, or we measured voltage as a function of time. Voltage is measured as a function of time, something like that. And related, it's actually okay to use the first person. Some people are taught that you can't use the first person, I or we, but you actually can in scientific papers. Most everyone worked in a group, so you would say we. That helps get rid of the passive voice. And it's totally fine. As here, this is really vague, and I don't know, but it's kind of tough. You want to write as concise as possible. You want really short, like understandable sentences. You don't want it to be like a bullet point, but you also don't want it to be like a 14-page essay. You have to find how can you say what you want to say in the shortest amount of words possible. Scientific writing is really different than creative writing or anything else. like that. It's more about an economy of a language, and you're trying to communicate information to people, right? It's the same way science is about communication often, and this is what happened here. I say here, only give facts, not opinions. Don't tell me if the experiment was good or if you had a good time. You just tell me, what did you do? What did you get? What were the results? What do the results say? Let them speak for themselves. You can say, if you're doing an experiment where you're measuring a constant of nature and you can compare it to the known constant, you can just say, our value agreed with uncertainty, or our value didn't agree with uncertainty, and then you can tell me how you might improve it in the future. Last one here, I say data tables should be used sparingly. I gave you an example of a data table, and I always like to do that, but what ends up happening sometimes is people will get really excited about using a data table. Use them for summaries of results, but you don't use them for like raw data. Raw data, I think I had it somewhere else, but raw data doesn't show up in this part of the paper. The raw data would be like on a supplementary information, and that's actually what I have your lab notebooks for. So related to this, of course, there's a lab notebook due for this. Just follow the outline that I gave you before. You should be fine, but you need to include all your raw data in your lab notebook. Okay, so that should be, I think, enough instructions for everyone to get started on their first one. What I have here is an example of my first, not my first, but my kind of my main paper that I wrote in graduate school. It's about a decade ago. And it's about, I worked in biophysics, and so we worked on, well, you can read the title, Spatial Organization and Mechanical Properties of the Paracellular Matrix on chondrocytes. So you're not going to know what all those words mean, but you can see mechanical properties and spatial organization. I think you can understand that. That's the physics part. I'm in biophysics. And then no Copilot. And then the paracellular matrix of chondrocytes, that's the biology, right? So it's biophysics. This is actually, I didn't think about that ahead of time. It's a great title for this to explain what my field is. The paracellular matrix is basically a matrix, like a polymer network that exists on the outside of cells. So basically we were studying the mechanical properties of a polymer network that exists on cells. Chondrocytes are just the type of cell we use. Those are cartilage cells. They have big ones. You can see here is our list of names. I'm at the front because I was the first author. I was the person that did it. This is important for you guys, but then the last person listed in these is actually the professor of the group, the PI, the primary investigator. So that was my PI, the person who ran the lab. And the people in the middle are the people who helped, usually other grad students, etc. So here's the abstract. It's actually quite long. I think it's longer than the 200 pages or 200 words I told you. That's okay. It has a little bit of the information initially, like introduction, because it's biology or biophysics. You kind of have to explain what you're talking about because not everyone who reads this is going to know. They might know polymer physics, but they might not know anything about this. So you have to give them a little bit more context than you guys will need to do. And you can see towards the end here, it says with this result, we estimate the variation, blah, blah, blah. So here we're talking about the results of the work. So the introduction here, you can see the introduction immediately assumes I don't know what the paracellular matrix is. It says the paracellular matrix lies at the nebulous interface of the ECM and the cell surface. ECM is a different thing in biology people have heard about. And then paracellular matrix is, you can see this is where I'm using acronyms. Although in the abstract I had to define my acronym, I couldn't assume that they read the rest of it. I follow my own rules, that's great. You can see here all these references. This is because the introduction will have a ton of references to previous work. Obviously my group, this wasn't the first work we did on this thing, so this is previous work by other people, but also us. We talk here about the different biology, the different experiments we're going to do. You can actually see here, this is actually a figure for the introduction. This is a figure showing a cell. This is actually a rat cell right here. And it's surrounded by, these actually red blood cells from pigs that we actually use to do the assay. I'm not going to talk about the biology here. It's kind of interesting. And then there's actually, this is a diagram of the system. That's kind of interesting. You can see here's the figure. There's the figure caption. After that comes the experimental method section. It's actually broken up into multiple parts. You'll notice here I use subsections a couple of times. You may or may not be valuable for you guys. So cell culture, that's how we grew the cells. Passivation of microspheres is how we prepared the microspheres. We're going to do this like the chemistry. It's like the biology section, that's the chemistry section. And the rest of it's like the physics. Particle exclusion assay, optical trapping. This is actually involving optical trapping is what I did. That's why it's in physics. And the assay is actually the experimental assay. Like telling what an experiment did. Oh, we took this bead and we pushed stuff in, blah, blah, blah. You can see here there's actually no theory section. in this field, you wouldn't have a separate theory section. It kind of depends on the specific field. For us, we'll do one. And then there's the results. So then we say the optical force probe assay introduced in this work interrogates by telling me what we're doing. And then immediately we have a data plot, and it's showing you, this is actually the force, piconewton force, over time pushing a bead into this invisible network that you can see that's surrounding the cell. So basically I'm pushing a bead into this thing and watching the force push back on it. And this is the force assay looking at piconewtons. Yeah, and the rest of it here, we talk about all these different plots. You can see, here's some plots. You can see this first one doesn't have a legend. The second one does because I've pushed 4 times in a row and I get the same result. This one here has a legend because there's 2 different beads that we push into it. I'm not going to be so anal about this, but you can see here, these are designated by color and shape of the icon because you have to assume people are going to print your paper out in black and white and read it. So you have to make it readable in black and white. You can see here, so the results as like a kind of a main section, and then there's like subsections. So this is, they have a malleable and robust paracellular matrix, and then I have dynamic force probe. So this is a different section where we did a different experiment that's shown here. This is a subplot where you have 4 plots in one plot. I can imagine certain experiments that we do that do this. I'm not going to require you make a sophisticated plot like that. That's okay. You can see actually here we have a table because it's the results. So you can see values plus or minus uncertainty. You can see the 1 significant figure for uncertainty and rounding. This one here has 2 sig figs on the 1, as I mentioned, as a possible rule you can do. And you can see all the data. There's all those numbers in here. And so this is all the results. The equations show up a little bit later when we're actually talking about analyzing the results of these. Your papers might be a little bit different, but you can see formatting the equations with references here. The first one actually starts and just says this thing explained by an exponential. Then we get a little bit crazier, talking about osmotic pressure, etc. Talking about correlation length, that's a concept in polymer physics. Anyway, this is a lot more results than you guys are going to have. And then this has results in discussion as separate. You don't need to do that. Just have them as one thing. Yours is not going to be so specific, but you can see the conclusion still is one paragraph that's kind of summarizing the results. And then, as I mentioned, the raw data is not here, but supporting figures, equations, one table, one movie, etc. Biophysics, you can have that kind of thing. Movies, which are cool, they're actually here, which is nice. And then all the references show up at the end. 62 of them or so, which is quite a lot. Okay, so yours is not going to be quite this long, but just to give you an idea, that's how I do it. So, yeah, so hopefully I've given you enough to think about and write the paper. This is I mentioned it in the document. This is difficult to do initially. I've tried to do this many different times, and I find the best way to do this is just to have you do it initially, and then I'll give you feedback, and then we'll kind of work on it through the semester as you write a second paper and then revise this one again. I hope that was helpful, and happy writing.